Hello, everyone. Welcome to today's live broadcast titled Discover Potentially Predictive Immuno Oncology Biomarkers Using Next-Gen Sequencing, presented by Fiona Hyland and Carl Morrison, MD, DVM. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by the Clinical Sequencing Division of Thermo Fisher Scientific. The Clinical Sequencing Division provides end-to-end next-generation sequencing solutions powered by ion Torrent semiconductor sequencing technology. Adopted by thousands of labs across the world and cited in more than 3,000 publications since its launch 6 years ago, ion Torrent NGS solutions are simple, fast, and flexible, making it an ideal choice for clinical research labs across the world. I'm Julie Simmeroff of LabRoots, and I'll be your moderator for today's event. Before we start the presentation, we have a few important announcements to make. We welcome your questions during the webinar. Since attendees will be muted during the presentation, please submit any questions by typing the questions into the questions box, which may be located near the bottom of your control panel. Following the presentation, we will address any questions that were submitted in the order in which they were received. If we run over the hour allotted and do not address your question, we will personally answer your question by email following the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now, I would like to introduce Fiona Hyland. Fiona Hyland is the Director of Informatics R&D at Thermo Fisher Scientific. Fiona majored in genetics as an undergraduate in Trinity College, Dublin, and in quantitative genetics with minors in statistics and population genetics as a graduate student at Cornell University. At Thermo Fisher Scientific, Fiona's teams have launched tools to help interpret sequencing data in clinical research contexts, including algorithms for tumor normal analysis, genetic disease, gene fusion, and genetic annotation. Please join me in welcoming Fiona Hyland.
Hello, everybody. I am going to talk to you today about the Oncomine Immune Response Research Assay and, more generally, our multidimensional approach to discovering and using biomarkers for immunotherapy in oncology research. So we all know that immune therapies are revolutionizing oncology research. We have checkpoint blockades, a number of drugs, anti-PD-1, anti-PD-L1, and IPI have been approved. Therapeutic vaccines are another approach, and there are adoptive T-cell therapies, CAR T-cell therapies, and many, many more options are in clinical trials. In fact, there are more than 800 active clinical trials ongoing for single or combination immuno-oncology. And the reason for this work is simple. Immunotherapy is providing the promise of complete and persistent response in a way that was not seen with traditional therapies or targeted therapies, where typically resistance would develop after some period of time. And in some proportion of individuals, persistent response is visible with immune therapies. And so the potential is enormous. But the challenge is understanding the underlying mechanism of action and identifying biomarkers for understanding that mechanism of action and for predicting response to immunotherapies, for clinical trials, for identification of appropriate therapies, for identification of appropriate combination therapies. And so this challenge, we believe, demands a multivariate approach. So currently, what is it that makes stratification a challenge? We don't have complete knowledge of the mechanism of action. There are properties of the tumor and properties of the immune system and of the immune cells that are very important, but they're not all fully understood, and their interactions are not all fully understood. There are PD-1 and PD-L1 assays, but they're not entirely sufficient for predicting response. IHC assays against PD-L1 are quite standard, but are not as reproducible as might be desirable. And so we think that there's a grave need to identify new predictive biomarkers to help identify patients who may respond to either single or combination immunotherapy, and indeed also to identify patients who may suffer adverse events so that they can be avoided. So the tumor microenvironment, the properties of the tumor microenvironment are known to be extremely important in understanding mechanism of action and in predicting response to any particular therapy or combination of therapies. And we expect that it will not be sufficient to measure just one or a small number of properties of the tumor microenvironment. Instead, we expect that it will be necessary to measure a broad range of signals that identify the properties of the tumor cells themselves and the properties of the various immune cells, with the goal of segmenting individuals more likely to respond or less likely to respond, as well as answer other important questions. And so, in response to this need for understanding at a research level what are the properties of the immune system that make patients uniquely susceptible to certain types of immunotherapies, we have developed the Oncomine Immune Response Research Assay. The Oncomine Immune Response Research Assay is a next-generation DNA sequencing assay. It is one of a family of assays that we have successfully launched and have been deployed all over the world and are in wide use. The common thread in all of these assays, in all of our AmpliSeq and Oncomine assays, are RNA extraction from FFPE samples for RNA assays, templating and sequencing, and these are identical for all of our assays. And then this assay, along with our other assays, has specific designs to enrich for the targets that we're most interested in and for analyzing the data. So we use the Ion AmpliSeq library creation technique, which has very many desirable properties. It works on very low amounts of input material, DNA or RNA, as little as 10 nanograms of RNA. It is compatible with FFPE samples, and it is highly sensitive to low-expressed genes. It has a broad dynamic range and excellent reproducibility. The kit includes also the analysis. So our goal is to make the analysis simple, and it includes panels and reagents for library construction in manual or automated mode. The panel consists of assays to measure gene expression of about 400 genes in an FFPE sample to identify the tumor microenvironment. We curated these 400 genes from a number of sources. We appreciate the suggestions and consulting from our collaborators, including National Cancer Center Japan, various pharma collaborators, a range of other Oncomine key opinion leaders, and in addition sources such as the annotation databases like David, the Oncomine knowledge base, clinicaltrials.gov, and of course a careful reading of the current literature. And so we finally ended up with 395 genes, including interferon signaling genes, cytokine signaling genes, T and B cell signaling genes, markers of lymphocyte subsets, immune checkpoint pathways and targets, tumor markers, proliferation markers, and a background of housekeeping genes. And these genes are chosen to support various types of experiments, method of action studies, biomarker identification research, and drug combination research studies. More specifically, we have annotated our genes into 36 functional annotations. These annotations are available to users of the panel, and they include a number of genes involved in lymphocyte regulation, involved in cytokine signaling, and you can see the details here, involved in immune cell markers, involved in checkpoint pathways, involved in tumor characterization, and we have a number of genes that are involved in all of these pathways and that are expected to be mutually correlated. So I'm going to show you some technical data first. So one of the very intense focus areas that we do when we develop a panel is we focus a lot on the technical performance of an assay, and we believe that having robust technical performance is very important to further enable biological and clinical research questions. This is an example of some data on various samples. The top row is the technical concordance reproducibility between fresh frozen samples. The second row is the technical reproducibility between FFPE samples. And the third row is the technical reproducibility between an FFPE sample and its matched fresh frozen sample. And as you can see, concordance is very high in all of these cases. And we compare that to the concordance between two different samples in this box. And you can see that, as one would expect, the concordance is much lower, indicating that the variance associated with technical reproducibility is far lower than the variance associated with a biological signal, confirming that the assay can be used for measuring biological signal. Another kind of technical validation and verification that we perform is concordance with orthogonal technologies. And so what we want to see is that the gene expression measurements that we observe with our assay, the next generation sequencing assay based on AmpliSeq technology, is consistent with measurements of those same genes with a different technology. In this context, we use qPCR as the orthogonal technology, which is widely recognized as a gold standard. And these are examples of the concordance between those two technologies on 8 non-small cell lung cancer FFPE samples and 2 controls. And this is absolute expression. So these plots look very nice. They look like they have high concordance. You might be used to seeing this kind of concordance for relative expression, but not for absolute expression, which typically different technologies give fairly different answers. What we observe here is we observe very high concordance between the qPCR on the x-axis and the Oncomine immune response assay on the y-axis. And this is true across the various FFPE samples for all of these genes and also for the controls. Another technical study that we did was a dilution series. So we took 2 samples expected to have different levels of gene expression for these 400 genes and did a serial dilution. We diluted them into each other so that the samples were present at different ratios. And from that study, we can predict the relative and the absolute differences in expression between the different samples of the dilution series. And so this plot shows you on the left the absolute fold change estimates observed versus expected, and on the right, the relative fold change estimates observed versus expected. And what you can see is that we see strong linearity, we see high dynamic range, we see low variability, and most importantly, we see that these desirable properties are true for genes that are high expressors, medium expressors, and also low expressors. So we've colored the various genes by their expression levels. Red is a high expressor, and the green color are low expressors. And you can see that this pattern of linearity, high correlation with the expected values, and large dynamic range are true equally for the low expressors as for the high expressors. This is very important because low expressors include very many genes of extreme importance for clinical research, and we want to make sure that the performance of our assay is very robust for the low expressing genes as well as the higher expressing genes. In this plot, we show examples of a number of different samples, 30 different non-small cell lung carcinoma FFPE samples. The left plot shows the expression level of 3 genes, and you can see that these genes are correlated with each other, as one might expect. These are CD8 plus T cell markers, and what we would expect is that for samples having enriched CD8 cells, we would see high expression, and for those not enriched with CD8 plus T cells, we would see low expression, and that indeed is what we see. On the right-hand side, this graph shows the correlation of markers for killer T cells across these same 30 FFPE non-small cell lung carcinoma samples. And again, you can see that the genes that you expect to be mutually correlated with each other for killer T cells indeed are strongly mutually correlated with each other. And so this exemplifies the value of a panel-based approach with a large number of markers. Rather than having to make one measurement on one marker and relying on one value, you can make measurements on a large number of genes that are expected to be highly mutually correlated, and all of that data reinforces each other, and indeed this is what we see. Once we were satisfied with the technical performance of the assay, we wanted to explore its properties on some samples. This is an example of the utility of the panel to stratify samples by TIL levels. Because of the content of the panel, we expect that we will be able to identify samples with high TIL and differentiate those from samples with low TIL. You can see that the high TIL samples are marked in red. We have technical replicates, 3 technical replicates, and first you can see that the technical replicates all cluster very tightly together, which is good and what we want to see. And in addition, you can see that the high TIL samples also cluster together within cell type. Again, this is exactly what we want to see. We want to be able to differentiate high TIL from low TIL samples, and we've demonstrated that the assay has the capacity to do this. We compared the assay with another similar assay, the Nanostring. We designed an experiment to study these 10 samples, different kinds of samples, breast, head and neck melanoma, non-small cell and renal cell carcinoma. Nanostring was performed by a third-party CRO that runs Nanostring routinely, and we performed the immune response assay. And what we observed was that the distribution of expression of genes in the panel was nicely bimodal in the Oncomine immune response panel, but not so much with the Nanostring panel. So each one of these lines is a single sample. representing the expression of all of the genes in the panel for that sample. The blue lines show the Oncomine immune response assay. The black lines show Nanostring. What we want to see is a bimodal distribution, 2 peaks, that are distinct from each other. The left peak is genes that are not expressed, centered at 0, and the right peak is genes that are expressed. And what this bimodal distribution clearly demonstrates is the ability of the assay to distinguish genes that are expressed from genes that are not expressed. So you don't want to have noise, background noise at the low end, impede the ability of the assay to identify expression or non-expression for low expressed genes, or to impede the ability to distinguish between lower and higher expression of low expressed genes. The Oncomine immune response assay clearly has these 2 peaks, this bimodal distribution, and in addition has a higher dynamic range, whereas the Nanostring assay does not have such a clear bimodal distribution, indicating that it's more challenging to distinguish non-expressed genes from low-expressing genes. In addition, we can see that the Oncomine immune response assay offers higher sensitivity on genes of interest. So the blue is the immune response assay, the gray is the Nanostring, and you can see that the immune response assay has higher sensitivity. Again, this is most important for low expressing genes, where you want to be able to distinguish expression from non-expression and changes in expression values. And finally, we used the assay to distinguish these different types of tumor samples to confirm that indeed we were able to perform stratification by clustering and by tumor type. What we found with the Oncomine immune response assay is that the algorithm was able to clearly cluster the different sample types, whereas in Nanostring we didn't see quite such a clean ability to distinguish the sample types, reflecting the perhaps the content and the performance, relatively speaking, of the 2 assays. In summary, the Oncomine immune response assay is a robust, sensitive, simple assay to run. We've built in an analysis solution where we take in the aligned bands and the target region bed files into a software suite that comes with the instrument, the immune response analysis plugin, and we generate summary statistics, QC plots, expression and normalized expression counts. Those are available from the analysis solution, and they plug in to third-party analysis tools to perform differential expression. In summary, we're offering a highly curated panel with content to assess the tumor microenvironment and identify predictive markers for drug response. It is optimized for FFPE samples and also works on other kinds of samples with low input requirement, as little as 10 nanograms of total RNA. It has high sensitivity to low-expressing genes, such as the interferon family, interferon gamma, and has a broad dynamic range with high reproducibility, automated next-generation sequencing workflow that is simple to operate, simple to analyze, with a fast turnaround time. Thank you very much.
Thank you, Fiona. Now I'd like to introduce Dr. Carl Morrison. Dr. Morrison is the President, Chief Scientific Officer, and founder of OmniSeq Precision Medicine Technology. He developed the vision for OmniSeq while leading the Center for Personalized Medicine and Division of Molecular Pathology at Roswell Park Cancer Institute. Dr. Morrison remains a professor and program member in cancer genetics at Roswell Park Cancer Institute. He was recently awarded the Thomas B. Tomasi Hope Award. This award is the highest honor Roswell Park bestows to the faculty member whose work has made significant advances in the fight against cancer while bringing hope to cancer patients. Dr. Morrison received his doctorate in veterinary medicine from The Ohio State University College of Veterinary Medicine, followed by his doctorate in medicine from the University of Kentucky. He completed his residency at the Ohio State University Medical Center. Please join me in welcoming Dr. Carl Morrison.
Thank you very much for being on today. I'm going to follow up very much with what Fiona was saying about technical performance, and these slides that we're going to be covering will be very much in line with how we have validated this assay in our laboratory. So I have two disclaimers here. You can go back and look at yourself, but they're just about the use of this assay with Thermo Fisher. What I really wanted to get into is how we're using this assay for really providing precision immunotherapy. We have been talking about, precision medicine for targeted therapy research and mutational profiling. There is, the personalized approach for targeted therapy, and the way that OmniSeq is using the Oncomine Immune Response Research Assay is to develop a personalized approach for immunotherapy. And there's at least 4 buckets that you can put this precision immunotherapy into. And the good part about the Oncomine Immune Response Assay, it covers at least 3 of these 4 buckets, which is T-cell receptor signaling, which is certainly one of the most important things because it's directly related to checkpoint inhibition. Something we call PD-L1 signature, which is kind of a subset of T-cell receptor signaling, and you can get the number and the, not necessarily the pattern of immune cells, but you can tell what is the contexture of the different immune cells in the tumor microenvironment. So I think we've all seen this picture, a very famous picture by Genentech that describes the immune life cycle. And many people have focused on this, the first step in this immune life cycle, or that is the destruction of neoplastic cells and the release of neoantigens, and that has been described pretty much as the mutational burden. The problem with the mutational burden is approach for precision immunotherapy. It's just one part of this entire immune cycle, and that's the way we've used this immune response research assay. Is we measure mutational burden with a separate assay, which I'll discuss in a moment. But then we use the immune response research assay to fill in all the other various forms of this cycle, of the immune cycle. So more specifically, what we do is we take the Oncomine Immune Response Research Assay and combine that with the comprehensive cancer panel that Thermo Fisher also offers for mutation burden. Run that on the Torrent S5 XL. We're running up to 16 samples per run and test controls. Our DNA is going from 10 to 30 nanograms, RNA from 2 to 10 nanograms. And we're not doing confirmations on any aspect of the testing. The entire process is run on the suite of Ion Torrent products, such as the Iron Chef, as I mentioned, the S5 XL, and the Ion Torrent Suite server. But what I want to be more specific about was, is how we measure the technical performance in our laboratory. And our laboratory is a New York State laboratory, so we're held to very high parameters for technical performance. I typically break this part down into 3 buckets: pre-analytical, analytical, and reproducibility. I've listed some of the various things here that we want, like under pre-analytical, what's the nucleic acid stability in sample form? In other words, is someone sending you unstained slides, or are they sending you a block, and how do you test that? RNA stability and FF versus FFPE. Thermo Fisher's claim is this is effective for FFPE as FF, but I think in every laboratory you need to prove that for yourself because every FFPE specimen has different characteristics. Dilution with adjacent normal tissue, addition of adjacent necrotic tissue, all these things that could impact the assay on pre-analytical. We'll touch on that, not every one of those things. Analytical, I think I will walk us through virtually every one of these. The nucleic acid input, these encompasses pipetting errors, genomic DNA contamination, these are errors that occur during the nucleic acid oscillation. Batch size, right? Does it matter if you run 2 samples or 16 samples? Do you get the same answer? Linearity of the process, right? Can you demonstrate that you can detect low expressors, moderate expressors, high expressors? I think Fiona really focused on that ability to identify low expressors, and I think that's a very important point that if you go look at whole transcriptome studies, you will see that whole transcriptome from fresh frozen tissue really does not work well at the low expressors. multiple reasons for that, but this assay seems to work extremely well at that point compared to whole transcriptome. And then there's the sensitivity of variant detection. You have to know what is your limit to detection. In other words, do you trust 10 normalized reads per million or 20 normalized reads per million? What is your cutoff for which you say this is just background noise? And then reproducibility, it's a very standardized process. Sometimes it's very difficult to present the data because there's so much data, and I'll show you how we present that data. We kind of move away a little bit into what we call the characteristics of the research specimens. I'm going to touch this. What is the phenotype that we're trying to identify, and how do all these different aspects influence the phenotype that you're trying to identify, such as the amount of stroma, the amount of tissue, the tumor microenvironment, what does it look like. So I'm going to start with the most straightforward of these, I think, that every laboratory needs to focus on, that is RNA stability and fresh frozen versus FFPE samples. We published this here very recently in Oncotarget using this particular assay. And we took a very, very stringent approach to this, and the stringency basically was on the specimens. So we took fresh frozen tissues that were large enough that we could bisect these fresh frozen tissues parallel to the longest axis. Then after they were bifurcated, we took ink and put on the face that was up, and the ink surface was mounted face up on a chuck to cut sections or curls for fresh frozen. And then the FFPE specimen went through processing, and then when it was mounted in paraffin, it was mounted with that ink spot up, so that we knew we were exactly comparing the same faces of one piece of frozen tissue versus the same section of FFPE tissue. And this shows 13 samples that we did in this study that we published. And as you can see, there was very good correlation between the FFPE and the FF. So this was our demonstration that FF was concordant with FFPE. The next two things were nucleic acid input and genomic DNA contamination. These are errors that can occur in your laboratory, and you often do not know that they're even occurring. How accurate are your pipetters, and how accurate are your pipettes? Are you getting genomic DNA contamination, and what's the impact to your assay? So in this example, you can see here we have a single sample that we compared doing the results. We really used about 50 genes here for this comparison to keep the noise down in the figure. But the assay was run with 2.5 nanograms of RNA and with 25 nanograms of RNA. And our recommended input is 10 nanograms of RNA. So we were on either extreme of a pipetting error. And as you can see, the concordance between a low RNA input and a high RNA input was very good. At the same instance, we did genomic DNA contamination, and we went all the way up to 50% genomic DNA contamination. And again, you can see that the correlation between presumably no DNA contamination and 50% DNA contamination was outstanding. If we move on to batch size is so important because most laboratories, you're going to run any type you do this assay, you're going to run it, probably once a week or once every other week, but you need to know, am I going to run this with 2 samples or 16 samples or 4 samples? And we demonstrated that it really didn't matter. It didn't matter how many samples you put in the assay because normalization corrected for the number of samples in the batch. So we're showing 1 sample here, and we're showing normalized reads per million, and on the x-axis we were showing it where it was run with 16 samples, and on the y-axis where it was run with 4 samples, and the concordance was quite good. The other thing is RNA-seq with the library dilution, right? What happens if you have a less than optimal sample and you have less than the recommended picomoles of library? So we took 1 library and diluted it. We ran it at our recommendation is 25, and we ran it at 50 picomoles, and we ran it at 1.56 picomoles. And you can see that the concordance was outstanding, so that you can allow for some pipetting errors and various things to occur around the library step and still get excellent results. The next thing, and we didn't do this just for 1 gene, but I did pick out a very common gene in immuno-oncology, PD-L1 or CD274, to demonstrate, right, that the detection of this gene was linear across a dynamic range. In this instance, we took 6 different samples, each of which had a different level of expression of PD-L1, and then we serially diluted the samples with each other. And as you can see, at this point, this is absolute reads. It's not normalized reads, because if it was normalized, these plots, since they are highly concordant, would become horizontal. But as you can see, the expression across the dilutions was outstanding. And when you get to the very lower limits, you can see here that we get somewhere down around 20 reads or so, and we get into the noise where we can no longer detect it. But outstanding results for PD-L1. We're moving on into this reproducibility. Anytime we do assay development at our site, we always have a positive sensitivity control, and we make sure we run that positive sensitivity control in every run. So we experimented, for our positive sensitivity control, we experimented with many different cell lines to find one that highly expressed the majority of the genes that are in this panel. So we tested several cell lines. We always take the cell lines and turn them into a pelletized form and then take them through a processor and treat them just like any other FFPE samples. And as you can see, this is the for this particular cell line that we eventually chose as our positive sensitivity control. We ran it for 43 different runs, and the correlation across all the different runs was just outstanding. And I was talking about reproducibility, so there's intra-run, between technician, between days, between barcodes. We didn't do between instruments here because at that time we were running all this on one S5 XL. But as you can see here, this is the way we always present our information in a graphical form. We take this information, put the detailed information over in Excel files, but this is easy for the reviewer or the viewer or anyone who wants to review your technical performance to see how we've done this. And you can see how we've had, different technicians, different days, different runs. And we used 3 different specimens plus a replicate of the 3rd specimen. And you can see the concordance is outstanding, and it's even really good for the replicates of the specimen 3. Then I'm not really going to go into clinical validity and clinical utility, but I did want to just kind of touch on, how do we know that what we're looking at is what we're looking for? So we had originally took an observation from whole transcriptome analysis looking for a particular phenotype, and on the right-hand side shows this phenotype from whole transcriptome analysis. And as you can see, on the right-hand side here, anything that's red is high expression, green is low expression. And you can quickly start to see that there's basically, high expressors, very low expressors, and moderate expressors. And that's what we used, that type of an analysis, high, low, and moderate, to say what is the influence of specimen characteristics on this pattern. And to do that, we did that primarily with principal component analysis. And we asked lots of questions about specimen characteristics. No way we can get through all of them today, but I think things you need to know about the immune phenotype is, does it matter what type of specimen you're testing? So, you can get a specimen that is essentially a cell block, so it's totally disaggregated. You really can't tell the tumor from the stroma, and sometimes it's difficult to recognize, what's the percentage of stroma and the percent tumor. We see these small needle core biopsies that are extremely small that get disrupted. They just look like little islands of tumor in a biopsy. Then we see some that are partially intact, and then we get research samples that are intact. And when we did our validation, we did this with several hundred samples. I think I'm showing 167 in this graph. And you can see here from principal component analysis in the first 2 dimensions, right, that there is no bias for any one of these types of specimen architectures to have influenced the signature that we were seeing among low, moderate, and high expressors. We looked at the same thing, and this was kind of probably a little bit of a surprising result to us. I was predicting that the amount of stroma that was in the sample would have an influence on the expression of these signatures. So we compared samples by pathology review. We marked the samples as abundant stroma, moderate stroma, modest stroma, or stroma that's really not apparent, almost like solid sheets of neoplastic cells. And principal component analysis, again, did not say that there was a remarkable difference in any of those characteristics. We looked at specimen size, right? We actually measured the specimen size, right, and put it into specimens that were less than 10 millimeters square, between 10 and 25, 25 to 100, and then greater than 100 millimeters square. So, you know, the latter two are kind of resection specimens. The 10 to 25 is typically a needle core, and a less than 10 millimeters, typically something that's like a needle core block that's been heavily used, right? So, again, we relied on principal component analysis and showed no difference in those specimen characteristics influencing the moderate or the high or the low expressors. This was probably one of the most interesting things that we did in this validation from a technical performance. And people always ask this question, right? What is the appropriate specimen to test for immunotherapy? Because sometimes a patient will have 2 specimens in an archive. It may have 3 or 4, maybe only 1. But what happens when you do that? So I apologize if this label's overlaying in the top half of this figure is what we call tumor-infiltrating lymphocyte score. And in the bottom half of this figure is what we refer to as the T-cell receptor score. But as you notice, there are 16 patients in this study, and the patients are listed along the bottom there. And then the results for one patient are listed vertically. And as you can see here, if we were to look here is one patient here that has 3 samples, and they were separated by approximately 2 years apart. And the T-cell receptor signaling score across all 3 samples was nearly identical. The same patient, when we looked at the TIL score, was highly concordant. And then we see some samples that are very concordant like this. And then we see some samples that are not so concordant. And like here is an example of a sample that was not so concordant. And roughly what we did when we got done with these 35 samples from these 16 patients, they were roughly divided into 2 groups. About half of them were highly concordant, like this patient and others that I've marked. And then there were about half of them that were like this patient or this patient, which are not as concordant. And it's the same thing for TILs or the T-cell receptor score. We at first thought this was chemotherapy effect, and we went and looked up chemotherapy for these samples, and there was no correlation. And if you really look down into details, what you will find here is that the most recent sample for all these patients except one that had a difference in the interpretation of the amount of T-cell receptor signaling or the amount of TILs, the more recent specimen was always the lower quantifiable sample. And it really came down to tumor burden, so the tumor burden is suppressing the immune system. It was a very exciting specimen study for us, but it all came back down to what is the appropriate specimen to test. The appropriate specimen to test is the most recent, because that's the one that's most in line with what the patient's current immune state is. This was our validation. There's quite an extensive validation. I certainly don't have time to touch upon lots of it, but you know, we've been really happy with this assay. We really like its performance among low expressors, and then anyone who's working in the immune response, we're chasing that interferon gamma signature, and it's just full of low expressors. And the assay does have cancer testis antigens type genes that are the off-on. So when they're on, they're extremely high, and when they're off, they're just there's hardly no expression at all. It performs just as well for the on-off signatures as it does for the low expression signatures. But it's really, I think its strength is in these low expression signatures and something you don't get out of whole transcriptomes. I do think that is all the slides, and I think it's time that we proceed to take questions.
Thank you, Dr. Morrison. Due to the technical difficulties, we will be addressing questions via email following the presentation. Again, we apologize for the technical difficulties, and we'd like to thank you for your patience. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through October 25th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. Thank you both once again. That's all for now. Thank you for joining us. We hope to see you again soon. Bye.